Okay, good evening, everyone. All right, everyone, switch on your videos. Let's start today's class. Everyone, I won't start the class until and unless all of your videos are on today.
Ma'am, just a minute.
Sure. You can take your time, but you have to switch on your videos. Yes, Khadija and Siddika, switch on your videos. Okay, Siddika, yours is on. Khadija, switch on your video. Am I audible, Khadija?
Yes.
Switch on your video. Also, make sure that no one switches off their video in the class. Okay. And yeah, so we had discussed about the chromosomal theory of inheritance. Did you people have any doubts in that? Okay. Now, today we're going to discuss a new topic, that is the linkage and recombination. Now, these are two terms that are, you can say, very closely related, but they do not, you can say, work as closely, but they are somehow related to each other. Now, talking about linkage. So, linkage refers to how, you can say, the genes are present on the chromosome. Like, what kind of association do they have? So, the way they are present on the chromosome physically, that offers their linkage. And again, linkage is different from, like, for all the different kinds of chromosome. Talking about the recombination, so it is the generation of non-parental gene combination. Now, do you people remember in meiosis, there was a time in prophase wherein crossing over took place? Do you people remember that? Right? So crossing over took place, and because of crossing over, what happens is that the genetic material gets exchanged. So the genetic, like this exchange of genetic material, it is known as recombination. This is the process wherein new characters will be exchanged between the parents. Clear? I mean, for the offspring. Okay, this much is clear. Okay.
Yes.
Now, these sex-linked genes were studied by Morgan. Thomas Hunt Morgan studied the genes that are like sex-linked genes. Now, four pairs were there, and out of those four pairs, like we know that Drosophila had four pairs of genes present in it, right? So, out of those pairs, three pairs were autosomes, meaning these are the body chromosomes, and then one pair was the sex chromosome. Same way, just like us, how many chromosomes do we have? Humans, how many chromosomes does a human have? Hmm? Khadija and Tamim, please switch on your videos. I won't ask it again and again. Please switch on your videos. Yes.
46, man.
46, meaning 23 pairs of chromosomes are there, right? Now, see, out of those, for example, if we are talking about the 23 chromosomes or the 46 chromosomes, now, out of the 23, the 22 pairs are like body chromosomes, like the normal chromosomes of a body. And out of this, one pair is the sex chromosome. Now, Morgan carried out a dihybrid cross in Drosophila. So he carried out a dihybrid cross. And what do you mean by a dihybrid cross, students? Hmm? What is a dihybrid cross? We studied it yesterday.
When two characters are taken into consideration,
When
or
two
two pairs.
characters are considered. Yes, very good. So whenever two characters are considered, so we call that type of a cross as a dihybrid cross. And when only one character is considered, we call it as a monohybrid cross. So Morgan carried out a dihybrid cross in the case of Drosophila, wherein what he did, he took one female Drosophila who had yellow body and white eyes, and then he crossed that female with a male Drosophila that had brown bodied and red eye. Now, the ratio that came out, it was not following the Mendelian ratio, that was the dihybrid cross ratio, that is 9 is to 3 is to 3 is to 1. So these, like this particular cross, this dihybrid cross of Drosophila, it did not follow the ratio of Mendel that it was supposed to follow. Now, there must be some reason. See, what happened is that this did not follow the ratio of Mendel because the genes were tightly linked. And whenever genes are tightly linked, they do not participate in recombination, meaning they don't get exchanged. Now, if genes are linked tightly, they show very low recombination. If genes are linked loosely, they show high recombination. Being linked tightly means that the genes on one chromosome are present very close to each other. And being arranged loosely means that they are present far apart from each other. Now, when the percentage of recombination was found out, so it was observed that for the tightly linked genes, the recombination percentage was 1.3%. Only 1.3% recombination took place. And when the loosely linked genes and their recombination percentage was calculated, so it was found that it was 37.2%. So obviously now you can see that there is a big, you can say drastic increase in the percentage of recombination. So, in a nutshell, linkage means how closely or what is the physical presence of the genes on a chromosome. Now, the genes can be tightly linked when they are very closely situated, and they can be loosely linked when they are situated far apart. And whereas recombination is the generation of non-parental characteristics, which happens by which process? So, it happens by crossing over. So we can mention it here that it takes place, it takes place during crossing over. Clear? Also, students, like how many tests have you had here in your online tuition? Like how many tests have you had? Hmm?
One. I did one for bio. Only
One.
one.
What about
Yeah.
I
others?
did. I did three. Yes, I didn't did the last one. I did the three
Hmm.
tests. Yeah.
So there was like in totality 4 tests that happened,
Yeah.
right?
Mm.
Yes,
Yeah.
ma'am.
Okay.
Yes, ma'am.
Okay, so like why like I yes yeah I saw your answer sheet. I have checked yours, but like most of the students I haven't received your answer sheet. So like did you people not take the test? Hmm?
No, ma'am, I didn't give any test.
You didn't? Who else did not?
Ma'am, yesterday was my fourth test.
Oh, you did give the test?
Yes, ma'am.
Okay, how did it go? How was the test?
Ma'am, it was good.
It was good?
Yes, ma'am.
Did you find the questions easy?
Yeah, ma'am, but the last one was a little bit difficult.
What was the question?
Ma'am, it was, I have not written in my notebook. It is in the PDF.
Okay, okay. Do you remember the question? Wait.
No, ma'am, it was very long question. Something F1
Oh, I get
generation.
it. Mm-hmm. I gave you a cross. Who else beside Ikra gave the test, like the previous test, which was like the syllabus was reproductive health and genetics? You did, Khadija? How
Yeah,
was the test?
it was fine.
The
I could
last,
have written so much more details in it, but I didn't. The last one, it was the cross. You had to draw the cross.
yeah, I gave the cross. So Ikra, you found the cross difficult? You did
Yeah,
not
ma'am, it was
understand?
a bit difficult. Yeah.
Okay, okay. And Khadija, what about you? The cross was easy or difficult? You are not unmute.
Oh, I need to practice it.
Okay. Anyway, students, we're going to discuss it once we are done with the chapters, and you people can come to me with all your doubts. Does someone have their TV on or something? Like I can hear.
Ma'am, I have a doubt.
Yes, yeah. Say
Even if I didn't read the test, can I still get the paper? I want to practice.
the question.
Yes, the question paper.
I think so. You can. I mean, I don't know how it goes along for your test. Like, how do you people get notified about the test? Where do you get the questions?
They send us through WhatsApp, ma'am.
They send you the PDF of
Yes,
the
ma'am. They
question.
Yeah,
send us the
ma'am.
yes, ma'am. They send us the question paper.
Okay. Okay. All right. So for those of you who haven't got like given the test, I can like provide you with the PDF. You can practice it. It's like good. And like mainly I've started to focus more on the subjective questions because that is what is mainly asked, right? I mean, please mute yourself. I don't know what voice is coming from your side.
Ma'am, our sir said that this time around they'll ask more MCQ questions.
In your school,
Yes.
same for everyone. Hmm? Or there is
I
like a ratio, like more like mixed of him.
no. He said in half yearly exam he'll ask 35 MCQs and 35 theory part.
Okay, so it's like the ratio is equal. Now see,
Yes.
as far as MCQs goes, like the basic concept is like there is one answer, you take that answer and you write that answer. But subjective, like how you answer your subjective questions, that tells me or that shows to me how well you've understood the topic. And again, you people are the senior most class, right? You're the like senior most in your school. So answer like that. Answer like you're the senior students. Okay. So yeah, I'll send you the PDF for all of those that haven't attempted the test, and you people can practice that. Okay. Now, okay, why did I start talking about the test? I don't know why did I start talking about the test. Anyway, but remember that the recombination takes place during crossing over. And where does crossing over take place, students? Crossing over takes place in
Recombination.
recombination takes place because of crossing over. And where does crossing over takes place? Hmm.
Chromosome.
Yeah, it takes
Kind.
place in chromosome, correct. But I am talking about the stage. The stage.
Prophase. Prophase 1.
Very good. Prophase 1. It takes place during meiosis, and more particularly, it takes place during prophase 1. Very good. So happy. Now, any doubts up until now? Okay. Recombination and linkage is clear to everyone? All right. Now see. There was a person whose name was Alfred Stewart Went, and he used the recombination frequencies to measure the distances between genes and map their positions on the chromosome. Depending upon how much recombination took place in between genes, he started to use that frequency to map where the chromosomes are present or where the genes are present on the chromosome. So when the mapping of prokaryotes is done, just like how we have SI units, right? We have centimeter, meter, like we have the units for measurement, isn't it? Kilometer is for distance, centimeter is for shorter, whatever you call it, that thing, measurement, right? Liters are there. So same way, when mapping of prokaryotes are done or prokaryotic organisms are done, then the unit that is used is map unit. And when the mapping of eukaryotes is done, then the unit uses centimorgan. Clear? Now, crossing over is the exchange of genetic material, and the exchange of genetic material is recombination. So if you say crossing over or if you say recombination, the thing is same. And see, 1% recombination, 1% recombination is equal to 1 map unit. So it tells us, like, 1% tells us the distance. So 1 map unit. Any doubts up until now? This chapter is that kind of a chapter which will not make a lot of sense because you cannot relate to it, right? There are many things that you cannot relate. So even if it doesn't make sense, think of it as a thing that you have to just mug it up. The best thing is, like, you try to understand the topic, you read and you read and you try to get the concept. Now it's my, like, personal advice to all of you that when you're not able to understand the topic, you cannot leave it, right? Please don't do the mistake of leaving the topic. I don't understand it, I won't study it at all. No, do the next thing, that is the best thing. Mug it up. Whatever is there, just read it and learn it. Do you know that term? It's like, actually it's Hindi. They say ratta maro, rat jao. You do that with, if you do not understand anything. If you can understand, then it's great. The first approach should be understanding the topic. If you don't understand, come to me. And even if I teach you and still you don't understand, then ratta mar jao. Okay? Because it's better to mug it up than to leave it. Okay. Now we're moving on to sex determination. Now, sex determination, how is it done in human students? Sex determination with the help of dash.
Chromosomes.
No, but like Indians, or not just Indians, in general, sex determination. How do we find out the sex of the fetus?
We use amino sen,
Amniocentesis. Very good. Amniocentesis. You remember the fluid, amniotic fluid is used to find out the sex of the gender. As I told you, I'll never not stop asking you the questions related to the other chapter. Anyway, moving on to sex determination. See, establishment of the sex of an individual through different ways is known as sex determination. This is your basic definition about sex determination. Now remember that the sex determination varies in different organisms. Like for us humans, we can know the sex of the, or you can say gender of the fetus through the chromosomal patterns, right? But there are some other organisms in which different methods are also used, such as in the case of turtles. The sex is determined by the incubation temperature, meaning how much of heat period the babies have gotten. Turtles are egg laying or birth giving organisms.
egg laying.
Egg laying, right? Everyone agrees? So you know that whenever any organism lays its egg, that egg has to be incubated. That is why birds sit on their eggs so that they can incubate them. Incubation is providing heat. So that heat, that temperature actually determines the sex in the case of turtles. Okay, it's very bizarre. Also, do you people know the difference between turtles and tortoise? Or are they the same thing?
Turtle, they swim in the water with their paddles, and tortoise, they have legs. They can
Very good. Exactly. The turtles are the ones that can actually swim, and the tortoise are the ones that can go under their shell. The turtles can't. The ones that can go under their carapace, the shell, are the tortoises. And turtles and tortoises belong to which group? They are what? Amphibians, mammals, what are they?
Amphibians.
Amphibians.
I just gave two things. I did not wanted to give the answer. These are not all the options. So don't choose in between them. Say something else. They are reptiles. Okay. Now see, in the case of Drosophila, birds, humans, butterflies, grasshoppers, and honeybees, sex is determined by the genes. With the help of genes, the sex can be determined. What is the common name of Drosophila? Common name of Drosophila?
Fruit fly?
Fruit fly. Okay. What is the scientific name of humans?
Homo sapiens.
Sapiens.
What is the scientific name of honeybees?
Mm,
It is Apis indica. Okay, now see, in all of these 6 organisms, the sex is determined by the genes. Okay, now there was a person whose surname was Henking, and he in the year 1891 discovered the X bodies. More particularly, he discovered the X chromosomes. Okay, and then Stevens in the year 1902 discovered the Y chromosomes. And then Wilson and Stevens together, they said that X and Y are the sex chromosomes. They are responsible for the sex of the fetus or the organism, meaning they do not participate in the bodily functions. Now, there is a term that you should know, that is heterogamety. Now, if you break the word, hetero means what, students? Hetero?
different.
Different. Very good. Different. And gamete means?
Gametes.
Gametes?
Yeah, sort of related to the gametes, production of the gametes. Now, heterogamety can be found in males and it can be found in females also. Now, see, for example, what is the chromosome of human male?
XY.
XY?
Yeah, human male has XY, right? Everyone agrees with this?
Yes, ma'am.
And human female?
X, X,
XX.
X square.
This is the case of male heterogamety. This is the case of female homogamety. Female here in the case of males produces only one type of gametes, and that is known as homogamous. And male in the case of humans, they produce two different kinds of gametes. It can either produce X or it can either produce Y, and that is heterogamety. Clear? Now this heterogamety of male is found in humans, but in other organisms, somewhere females are also heterogamy. Now let us see how. First, we'll discuss the case of male heterogamety. In this type, the male individual produces two different types of gametes. In this case, the sperm would determine the sex of the offspring, meaning the male gamete is going to finally determine the sex of the offspring. If it gives the X chromosome and that fuses with the X of the female, then that becomes a girl child. If it gives the Y chromosome and that fuses with the X chromosome that the female produces, it will become a male child. So now there are two mechanisms under the male heterogamety. The first is XO type and then there is XY type. Now first we'll discuss this thing that we already know. XY type of male heterogamety is found in Drosophila and in the case of humans. Here, and all the mammals mostly. And here what happens? The male produces XY chromosome. The female produces XX chromosome. So in this case of humans, 23 pairs of chromosomes are there. Out of these 23, 22 pairs are autosomes. Autosomes means they are body chromosomes. And then one pair is allosome. Allosome means they are sex chromosomes. as I told you before also. So this is how basically the whole entire organization of chromosome is like. Now, if there is 22 autosome plus one X chromosome, and there can be 22 autosome plus one Y chromosome. So this is the case of a sperm. The sperm which is fertilizing the ova, that can have 22 plus X, or it can have 22 plus Y. But the female will have 22 plus X in all the cases. So this is the case of male heterogamety, and under that it is XY type. Remember the example that XY type is found in Drosophila and mammals. Any doubts?
No, ma'am.
Sure? Clear to everyone? All right. Now coming on to XO type. Here, this type of sex determination is of grasshoppers. Here, the male grasshoppers have XO chromosomes and the female has XX chromosomes. Okay, now again, this is just like how there is in the case of mammals and Drosophila here, but AA is the autosome, meaning the body chromosome, and XO is the allosome. Now, this male can produce X chromosome or it can produce O, and the female will produce X and X. And when the fusion will take place, so if the X of the sperm fuses with the X of the ova, it becomes a female. If the X of the sperm fuses with X of the ova, again, it becomes a female. But if the O fuses with the X, then there is a male, and same case. So XO, it's just the change of an alphabet, but the whole entire concept is male heterogamety. Is it clear to everyone? Male heterogamety is clear? All right. Now moving on to female heterogamety. Now, just like in the previous case, the male was producing two different types of gametes. Here, the female is going to produce two different types of gametes. Now, under that, there are two types. The first one is the ZW type. Here, the female produces Z and W type, and the male is ZZ. So, this is found in the case of birds. And ZO type, here the female, again just like how it was there in the case above, here ZO type is the female and ZZ is produced by the male, and this is found in butterflies. The whole entire inheritance will be the same, crossing will be the same, it's just that the name of the chromosome is different. So female heterogamety is clear to everyone? Any doubts?
No.
I can explain it again. Female heterogamity.
See, female heterogamety is the case wherein a female produces two different types of gametes. Under that, there are two conditions. Just like how in the previous one, there was XY and XO type. Here, there is ZW and ZO type. In both the cases, the two different kinds of gametes will be produced by the female. So, ZW type of female heterogamety is found in birds. In the case of birds, the female is responsible for the sex of the bird, baby bird, or whatever it is. And then in the case of butterflies also, the female is responsible, but here ZO type of gametes are there. Clear? Clear to everyone? Any doubts? Sure?
No.
All right, very good. Now we're going to talk about the sex determination in the case of honeybees. Can you see my screen? Or have I written just this much only? No, yes. I got scared for a minute. All right. So talking about sex determination in the honeybee. Now this is the most interesting. It is the most interesting out of all. Now here, the sex determination in the case of honeybees is based on the number of sets of chromosome an individual receives. Now what happens in the case of female honeybees, there are two types of honey. One is the queen bee and one is the worker bee. The queen bee is, you can say, the queen. Obviously, the worker is the one that makes the nest, the honeycomb and everything. That is the function of the worker bees. And then the male is just one type. It is drone. It is known as a drone. These are very tiny. If you look at a drone and if you compare it with a queen or a worker, the drones are very tiny. Okay, so these are known as the classes of honeybees. Just like how in humans, you find one male human, one female human, and then there are transgenders also, right? But in the case of honeybees, you'd find queen, worker and drones. Clear? Now, what happens is that the queen or the worker is made as a result of fertilization. Fertilization, you understand? The fusion of sperm and ovum. So after the union or the fusion of sperm and ova, the offspring is a female. So whenever a sperm and an ova will fuse in the case of honeybees, the offspring will automatically be a female. Now that female can either be a queen or a worker, that depends, but it will always be a female. Never will it be a male. An offspring which develops from an unfertilized egg, the egg which is not fertilized, it did not undergo fertilization process, that becomes a male and which is a drone. So this is very peculiar type of sex determination, and that actually happens in nature. And males, or you can say male honeybees, are a result of, you can say they do not have fertilization. And this
Ma'am,
process,
how is the male born if the egg is unfertilized?
yeah, that egg develops into a male. There is no
Oh.
fertilization taking place. And the, okay, I have a great question that came to my mind. Okay. So
Oh,
when the egg does not get fertilized, that becomes a male. And when the egg does get fertilized, then that becomes either a queen or a worker. Now, the question, what will be the ploidy of a female honeybee and what will be the ploidy of a male honeybee? If you've understood what I've said in this whole entire thing, or in general, if you've understood human reproduction, sexual reproduction in flowering plants, you can answer my question. What will be the ploidy of female honeybee and male honey?
Female honeybee will be diploid and male honeybee will be haploid.
Very good. Exactly. And now, can you explain how?
because
Because
in
in
females, two
female,
gametes are
there are
fusing,
two gametes, yeah.
Yeah.
and in the
So
male, only
because
one
of,
gamete.
yes, because of the fusion of a sperm and an ova, n plus n is giving us 2n. So the female will be diploid. But talking about the male, it is developing from an unfertilized egg. What is the ploidy of egg? Egg is haploid. So this male will be haploid. Okay. Now, and also remember one more additional information that in totality, the honeybees have 32 chromosomes. Just like how we have 46 chromosomes in total. So they have 32 chromosomes. Now, this type of sex determination of honeybees is known as haplodiploid sex determination. Because the males are haploid, the females are diploid. So this is haplodiploid type of sex determination. Is this clear to everyone? Any doubts? Sure?
Yes. Ma'am, in this chapter, are we gonna learn how, like, gender, how the sex is determined for a tortoise or turtle?
No. That's all. Like that, I don't think even that line is there in your NCRT book. That's an additional line that I've given. Is it there? Can you check? Because I haven't like seen NCRT
One
with that.
minute, ma'am.
Just check it. I don't think it's there. And no, there is not much information about turtles and tortoises sex determination.
No, ma'am.
No? Okay.
No.
Anyway, let me show you the picture of the queen and the drone and the worker honeybee. Can you see my screen?
Yes, ma'am.
Yes, ma'am.
Okay, see this. The workers, they are tiny. Do you see how elongated the body of the queen is? It's very elongated, right? And how,
Yes.
see this thing wherein you can actually differentiate in between all of them. What is it known as? This phenomena where you can differentiate between a male and a female with respect to their external appearance. There is a
Okay.
particular name.
Sexual dimorphosis.
Yes, very good. I'm so happy that you answered it. Sexual dimorphism. Dimorphism means it has two different kinds of like shapes or structures or forms. Now here the workers, do you see they are teeny tiny, the drones, and you know what is the one sole purpose of drone? If you think about the life of a drone, you'll be so sad. They are just present to fertilize the queen. And after fertilizing the queen, they die. Really, they die after fertilizing the queen. They have no other purpose. The workers are there for making the honeycomb. The queen bee is there to make more workers and queen, right? But the drones are just there to fertilize the queen and then they die. They literally die as soon as the fertilization is done, they die. Clear? Any doubts?
No, ma'am.
All right. Okay, now you people can note down. Let me just get back to the screen.
Ma'am, we still have time, I think.
Hmm?
We still have time, I think.
Yeah, you do have time. I'm telling you to note down. I'm not ending the class right now.
I don't know, I wanted to hear my explanation.
Oh, no, no. I have too much fun with you people that I'd end the class so early. All right. Okay. Now you had written up till here, right, students? The
Yes,
chromosomal
ma'am.
theory of inheritance. Okay. Now you
We did
people can.
the like heredity test.
Hmm?
Yes, ma'am.
Okay. Like heredity. Okay.
Yeah.
Yes. Now you people can note down. You can switch off your videos, write comfortably.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes.
Yes, ma'am.
Ma'am, done.
Done, ma'am. Done, ma'am.
Ma'am, done.
Everyone's done?
Ma'am, 1 minute.
Okay, okay.
Done, ma'am.
Done, ma'am.
Ma'am, ma'am, done.
Everyone's done?
No, ma'am. 1 minute.
No, ma'am.
Okay, okay, waiting.
Done, ma'am.
Done, ma'am.
I'm done.
Ma'am, done.
Everyone's done?
Ma'am, what's the full name of Alfred? Son. Done. So, done, ma'am.
Done, everyone?
Ma'am, just a minute.
Okay, okay.
Ma'am, done. Ma'am,
Done, ma'am.
ma'am, done.
Everyone's done?
1 minute, ma'am.
Okay.
Done, ma'am. Done, ma'am.
Everyone's done?
Yes, ma'am.
Oh, okay, very good.
I'm done.
Everyone's done? Everyone's done?
Yes, ma'am.
Done, ma'am.
Are you done, students?
Yes, ma'am.
Ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am. Ma'am, done.
Done, ma'am.
Everyone's done?
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done.
Done, ma'am.
Everyone's done? All right, students. So with that being said, let's just keep it till here only for today's class. And in the next class, we'll start with a new topic, that is mutation. Okay.